Okay. Good afternoon, everyone, and welcome to Rutgers Day Vaccine Q&A with Dr. Shobha Swaminathan. My name is Jameer. I am a health educator with Rutgers New Jersey Medical School Research with a Heart, and I'm so excited to be able to moderate this wonderful discussion about vaccines and all the wonderful work we've been doing here at Research with a Heart. Thank you all for attending. And hopefully you have some really great questions for Dr. Swaminathan. And it is a couple of seconds from 1 p.m. So without further ado, I want to introduce our wonderful principal investigator and just let her lead the way on this fabulous Rutgers Day.
Thank you, Jamil, and welcome everyone. I'm so glad that you all took the time off from your Saturday to join us on this exciting discussion. Hopefully, I'm going to try to keep my presentation brief to try to allow for ample time for questions. So I just have a couple of slides that I'd like to share to get the discussion started. There we go. All right. So in an overview, so we will be trying to go through just a little bit about our team and what we do at New Jersey Medical School at Research with a Heart, and how we got into the COVID-19 vaccine clinical trials, followed by updates on some variants and the J&J vaccine, which I know everyone, or at least I have been following minute by minute, and concluding with some ongoing research activities at our unit. So who are we at Rutgers, New Jersey Medical School Research with a Heart? Well, we are a very cool and dynamic team. As you can see, Jameer is just one of our awesome team members. And this is just this picture reflects a small piece of our team because our team is currently almost tripled in size, in a large part because of the work that we've been doing over the past few years. And the alphabet soup, as I call it, that you see just represents the diversity of funding that we get, mostly from NIH. We get funding from the National Institutes of Health from multiple different departments to offer predominantly HIV treatment and prevention studies. But then in the past year, we literally had to drop everything we did and pivot to focus our attention on this on the COVID-19 global pandemic that has impacted every single one of us. And as a result of all of those, all of that work at our site and the hundreds of thousands of people across the world, we currently have 3 approved COVID-19 vaccines in the US. The 2 messenger RNA vaccines that you can see on the top are the Moderna and the Pfizer, and then the Janssen or the J&J vaccine, which is an adenovirus vector vaccine, and the only one with a single dose, right? So those are sort of the key points. These 2 are RNA. This is the adenovirus. Both of these are 2 doses. This is a single dose vaccine. And they're all approved under emergency use authorization. So what is a messenger RNA vaccine? It sounds kind of fancy. And I hear this term and people say, oh my God, that's something to do with genetics. They're going to change everything about me. I'm going to become an alien. Well, guess what? None of that is true. The advantage is that the messenger RNA kind of bypasses multiple different steps and really helps for early vaccine development. The science didn't just happen yesterday. Messenger RNA technology has been around for almost 20 years. We just didn't really have a pandemic to leverage the need to fast track that to a vaccine development. It does not integrate with your DNA. It doesn't even enter the nucleus. So it doesn't alter your genome. I've gotten the messenger RNA vaccine and I tell my patients, look, you see, I don't have any horns. I'm not growing any wings. I'm still the same person that you knew me. It also is interesting that the messenger RNA has a very short life. It disintegrates very soon after it gets into the body. So it's not even there long enough to cause a whole lot of problems. Both the Pfizer and Moderna target very similar parts of the virus and prevents the virus from entering the cell. And the next slide, so this slide shows how that actually works. I love the New York Times because sometimes I think the scientific journals delay publishing things, and New York Times is on it. So this is something I took from the Times, and it's a good simplistic representation of how the messenger RNA vaccines work. So this is the squiggly thing in the middle that you see is a messenger RNA particle that targets the spike protein on the virus. Everyone has seen pictures of the coronaviruses with the globs on top. That's what we call the spike. So this squiggly thing you see is the RNA that teaches your cell to develop antibodies against those spike proteins. But because this is very unstable, it needs this lipid coating to keep it stabilized so that it's able to get to the cells in your body that it needs to get to. Once it bumps into your host cell, right, it fuses and then it releases this messenger RNA inside the cytoplasm. And within the cytoplasm, it just basically says, okay, here we go. And their job is to read the messenger RNA, make the proteins. When the proteins are made, there's 3 of them, they combine to make this spike trimer, and then they just stick out. It's very simple, either all of it or parts of it. Now what happens? So when this vaccinated cell, when it goes over here, when this vaccinated cell comes in contact with parts of our immune system, like the B cells and the T cells, they say, oh, wait a minute, I see these things. My job is to produce antibodies against it. So the B cells produce antibodies against these spike proteins. And as you see, these antibodies are found and then they float around in your body waiting for the virus to come. The other key part of it is it also trains the T cell or the cellular immunity to be there. They are kind of like your reserve. And what happens is when the body recognizes that there is an infection with COVID-19, it not only produces a lot of these antibodies, but the T cells jump into high gear and they stimulate these B cells to start producing more and more of the antibodies. So there's 2 pieces of the immune system that really help protect you from getting infected. So how effective were these vaccines? Both Pfizer and Moderna were highly effective. I'd be pulling teeth if I said, well, one was 94%, the other was 95%. Bottom line, they were extremely effective. And how were they effective, from getting symptoms from COVID-19? What's also important that they were almost universally protective in preventing serious COVID-19, meaning deaths, hospitalizations, getting on a breathing machine, those things that really cause complications, they were even more effective. So that's reassuring. So if it's that effective, so what were the side effects like? Is it really terrible? Well, the side effects were what you would expect when you get a shot. Anybody, I bet you there's not a single person in this audience who loves to get shots, myself included. We do what we need to do to make sure that we can live long, healthy lives, okay? So what happens when you get a shot? It hurts, okay? Sometimes they can have some, pain, redness, or swelling. Most of the symptoms, although a lot of people had those symptoms, they were generally mild to moderate. They were very rare that it was severe. A small proportion of participants also had some systemic, like flu-like illness, some headache and fatigue and joint pains and muscle pains. I will tell you, and everybody is different. Older participants tended to have fewer side effects. When I got it, my husband, my son, and I were out for a day after the second shot. And then almost like the switch got turned off the next morning, it was like, oh, what was that? We feel completely fine. In general, these side effects do get better within 2 to 3 days for those that have them. Not everybody has them, but those that do, they generally resolve fairly quickly. So let's switch gears to talk about the J&J or the Janssen vaccine. That technology is slightly different from the messenger RNA vaccines that we talked about. And they use what's called a Trojan horse. So what do I mean by a Trojan horse? It's really a trick to get the information into the body. So what these vaccines use, they use a different virus. So remember, we said the messenger RNA has a messenger RNA with the lipid on top to get it into the body, right? So this one uses the similar technology and uses this viral antigen, but coats it using an adenovirus vector, which is like a common cold type of virus. And uses that because, and the reason they picked that is it's it's easy to develop it, and it's a robust virus, meaning it survives easily on room temperature, which makes it easier to implement it in larger scales because you don't need the minus 80 degrees freezer, minus 20 degrees freezer. It's easier to keep it like in doctor's office, like you would get a flu shot or something else, so that it makes it a lot easier for worldwide distribution, including remote areas. Because remember, this is affecting every corner of the world. So we need multiple different vaccines to be able to impact the global population. And there are multiple other vaccines that I'm not going to talk about, including the Russian, the Chinese vaccine, that are using similar technology. We also are conducting an HIV vaccine study using a very similar technology. So this technology that Janssen is using is not new. They use an adenovirus 26 vector. Vector is a mechanism to transport the viral antigen. And even though it's a human pathogen, it's extremely rare and there's little to pre-existing immunity. This technology, again, has been well established. It's been used for Ebola, Zika, HSV, and the HIV vaccine study that we are currently enrolling in, the Mosaico study. In fact, Ebola was actually FDA approved last year and got lost in the midst of the pandemic for the prevention of Ebola virus. So, this is not new. What did the Ensemble study or the J&J vaccine study show? Overall, it was 66% effective, but in the US, it was about 72% effective. That was to prevent all kinds of COVID infections with symptoms. It was 85% effective against severe COVID, meaning it was 85% effective in preventing people getting admitted in the hospital ICU. But ultimately, that was also extremely effective in preventing deaths from COVID-19. Okay, and I've heard people say, well, the J&J was only 66, 70%, the Moderna was 95%. The Moderna study was conducted in the US alone. The Janssen was conducted across the world, including South Africa, where there were those variants. Did it have something to do with the vaccine efficacy? I don't know, but they are looking at those data to get better answers. But I want people to remember, the J&J vaccine was also nearly 100% effective in preventing severe COVID and deaths, because ultimately that's the most critical endpoint we want to protect. And this is my favorite slide, because people have said the vaccines came too fast. You can't get it right so soon. Generally, you'd be right, okay, and I get that. And I say that because when my husband and I had this conversation about vaccine, I'm like, it's not going to be here for another 2 years. What are they talking about? But that was, we didn't know the extent of multiple different things. The technology, particularly the messenger RNA and the Janssen technology, had already been used for other viruses. So there was a lot of information already available. So we had the technology. You had information previous vaccines. Also, for the first time, we had a public-private partnership. What do I mean by that? Typically, when you have a vaccine study, let's talk about the HPV vaccine or the HIV vaccine, it's one company looking at its one product, going slowly from phase 1 to 2 to 3, and companies by default are beholden to their shareholders and are risk averse. Nobody wants to try something that will fail. So they take a lot of time. And then if anyone has worked in clinical trials, there is a lot of bureaucracy and admin time for people just waiting on reading things. So if you really say, okay, no, let's just get rid of the bureaucracy, let's cut to the chase. Let's look at the data and make decisions based on data, not, oh, well, that person's on vacation. The IRB is not going to meet for another 2 weeks. No, this is a global crisis. We are going to do whatever we can to ensure the safety of the public. All the protocols, every single protocol is available in the public domain. I challenge you, you can Google the Moderna protocol, you'll find it. It's public information. Companies never shared protocols. We all had to sign, agreement saying that you're not going to discuss the protocol. But public safety was overreaching all of those concerns. There's also the independent data safety and monitoring board that reviewed all of these safety reports from all the companies to ensure if there were any patterns that were observed in one vaccine, that they would look out for those in the other. And sadly, and then at the end, the vaccines really showed, because the number of infections, the Pfizer and Moderna clinical trials occurred August, September, October, November. So people were fully vaccinated by October. And then guess what happened in November and December? Thanksgiving and start of the Christmas holidays. What happened to the US pandemic? We had a steep increase in cases, exploded at the same time that we were studying vaccine efficacy. And then the short incubation period. If it takes like a month for you to get infected with the virus and then develop an infection, it's going to take 2 months to figure out if the medicine works. When you have an incubation period, meaning I got exposed on Saturday, I get symptoms on Tuesday or Wednesday, you can show in a very short time that whether the intervention worked or not. And The clinical trial population, most clinical trials were conducted with 3,000 participants or so because that's cost effective, because we all are, cost conscious. Money is no bargain. The US government said, we are going to invest billions because it's a public health crisis. So they invested and they took the financial risk across the board. And they just did not take one vaccine. They invested in multiple different technologies because in April of last year, we did not know which vaccine strategy would prove effective. And we had to save our best, hedge our bets across the board. So because of the incubation period, the number of infections, the clinical trial size, and ultimately it was the vaccine efficacy. If the Moderna vaccine was about 40%, 50% effective, it would have taken us a year and a half. So when you start the study, you have to project, how long it will take for you to prove that it's effective, and that depends on is it 95% effective, 80%. The more effective the vaccine, the less time you need to prove that it's effective. That's why both Pfizer and Moderna were able to prove, within the 2-month period after completion of study enrollment, that the vaccines were effective. But the FDA still required the mandated the 2-month minimum safety. And they came up, and I looked this up independent of the study as an investigator. Most vaccine side effects occur in the immediate period, 1 to 2 weeks after vaccination, max 4 to 6 weeks. It's extremely rare to have, long-term side effects beyond that point. So that's why the FDA was okay in saying that a vaccine that's 95% effective and can save so many lives at 2 months, if there are no safety concerns, the benefit outweighs any less than 0.1% chance of remote side effects long-term. And ultimately it was because of the investment, but that's really the environment and how these vaccines came about. We've talked about some of the common myths about the COVID vaccines, long-term side effects. We've had a lot of vaccines for a long time. And sometimes we are guilty of our own success because we've forgotten what it was like before we had effective vaccines. Almost vaccine side effects long-term are extremely rare. And even the side effects that were reported with autism with MMR have been debunked completely, that there is no evidence whatsoever. And I can say that as a clinician, but also as a mother with a child with autism, that no, vaccines don't cause autism. The vaccine will give you COVID-19. No, they can't, because you need all of the virus to cause the infection. These are just giving you small pieces of the protein or the RNA to teach the body to produce antibodies without giving you the infection. If you had COVID, you don't need to get vaccinated. That's not true, because we have had people with COVID in the past who developed COVID-19 later on. Secondly, there's many studies that have looked at the amount of antibodies that people develop once they recover from COVID-19 and compared those levels to the antibody levels among those that got vaccinated. People who had the vaccine had way better antibody titers than those who had previous COVID-19. So the level of protection you get from vaccination is much better than from natural infection. If you had health problems, you shouldn't get it. That's not true. The clinical trial did include multiple types of participants. It was fairly diverse. It also included people with HIV, and there were no serious side effects. It is possible that if you have liver transplant or other immune system problems, that it is possible that the efficacy of the vaccine may be slightly lower, but it probably would be much better than than getting nothing. And vaccines alter your DNA. I've showed you the vaccines, the DNA is inside the nucleus. The virus, the vaccines have nothing to do with that. They don't even go there. So the messenger RNA vaccines will not alter your DNA. You can stop wearing masks. No, you should not stop wearing masks for various reasons. Because while the vaccines are effective, we don't know clearly how they're going to be against the commonly affected variants. So I still urge people to continue to wear masks. I still say I wear a mask anytime I go outside. And the vaccines cause infertility? No. I'm actually not even sure how to disprove it, because there is no scientific basis for even having that. It's almost like saying vaccines will cause you to have car accidents or vaccines will cause you to have something. It's trying to debunk something without evidence for its very existence. So all I can say is that there's no basis for that, and it's false. Does it work against the variants? We think it retains some efficacy against the variants. We know both Moderna and vaccine had very good effectiveness against the B.1.1.7 variant or the UK variant. And the B.1.135 or the South African variant has many more mutations than in the UK variant. That being said, the vaccines still retain efficacy. And I can speak from personal experience. My father is currently sick in India with COVID-19. He was fully vaccinated. His physicians can't believe that he's a 77-year-old male with multiple medical problems. The municipality came to their house yesterday, took him to the hospital because they said they couldn't believe that a 77-year-old could stay home. Well, guess what? His CT scan was completely normal. His blood work is normal. He feels fine. He had fever for a day and then he's better. He probably is infected with one of the variants in India, and I'm hoping he'll continue to do well, but they can't believe that a 77-year-old test results and everything look as good as they did. The only reason I can sit here and have this conversation with every one of you today without panicking is because I feel that the vaccine is protecting him from severe COVID-19. So will we need a booster dose? Maybe. We have to see what happens over time. Both Moderna and Pfizer are conducting studies specifically targeting the South African and other variants. If it appears that we do need to do a booster vaccine, just like we do a booster with the flu shot, maybe. But those data are coming, and we are waiting to see what happens. And lastly, a word on the pause on the J&J vaccine, because there have been a lot of concerns. So what happened? The J&J vaccine, as the CDC has been collecting data, so this is a sign that the system worked the way it should. Because when you vaccinate like millions of people, you might pick up things that occur 1 in a million or 1 in 2 million. There were a total of 15 reports of essentially types of blood clots in the brain out of 8 million doses. So again, that gives you a sense as to how rare that is. And it was somewhat similar to side effects observed in the European Union with the AstraZeneca vaccine. Interestingly, most cases were reported in women 18 to 48 years old. There was only one reported in a male. These have not been observed with either of the messenger RNA vaccines. So there was a 10-day pause from April 13th to yesterday until the CDC met to figure out does the benefit outweigh the risk. So ultimately what they determined that the J&J vaccines were saving so many more lives that they would because of the rare cause of thrombosis. So they recommended, lifting the pause to really recommend continuing J&J vaccine. They will probably give a warning for women under 50 to see if there's other treatment options available, then other vaccines available, then to consider those because the messenger RNA vaccines haven't had these side effects observed. And that's really important for us to know because we are not living in a vacuum. We're not giving COVID-19 vaccines just because we want to. We are doing that because we want to protect people from dying from COVID-19 because and it's everywhere. COVID is, there's a lot of asymptomatic shedding. So we can't really insulate ourselves in a pod unless we live in the middle of an island and just live off whatever grows there. So therefore, I think the benefits of these vaccines far outweigh the potential risks. So how did all of these clinical trials, come about, and what did we at New Jersey Medical School contribute? So this is just a quick snapshot. I'm not going to get into each one of those studies, but what you should know is that the COVID-19 Prevention Network, or the COVPN, was a cluster of sites that was put together very quickly because these were sites that were experienced in conducting clinical trials for HIV and other infectious diseases. And we directly pivoted. We dropped everything and we said, we have to focus on this. And we developed this. And we conducted the Moderna vaccine study and many other treatment studies for treatment and prevention studies for COVID-19, including this homebody study that looked at monoclonal antibody infusion among household contacts that was published about 2 weeks ago and shown to be extremely effective in preventing household transmission of COVID-19. And the last study we are enrolling is in case you see us in the community, is the COMPASS study, which is looking at community-wide seroprevalence of SARS-CoV-2 in health centers, nursing homes, and various community locations. What are some of our ongoing HIV treatment and prevention studies? We have 3 exciting prevention studies that we have. One of it is the Mosaico, or the HIV vaccine study, which is a phase 3, that's a big deal. That's one of the fewer phase 3 HIV vaccine studies to prevent HIV infection among gay men and trans women. We also have 2 new awesome studies sponsored by Merck comparing just pills. So remember, I bring the analogy of HIV prevention similar to oral contraceptive. No one size fits all. We need to have multiple different options. So yes, we have 2 pills, Truvada and Descovy, to prevent HIV. Cabotegravir, the injectable, will get approved. But then, it would be great to have a vaccine. And then the Merck study is looking at a once-a-month pill compared to daily Truvada among both women and gay men and trans women. We have multiple other studies for persons with HIV infection that I'm not going to get into. And I want to conclude with a huge shout out to my amazing team, because without my team, we would not be anywhere. The CoVPN, our research community advisory board, our community partners, and most of all of us owe a big debt of gratitude to the 100,000 plus participants who have stepped up and volunteered to participate in these and other clinical trials. These are our social media handle in Research with a Heart. I will conclude by I will never forget a quote one of my Moderna study participants said. He was an older man who said, I asked him, why did you do this? Because he said, I'm going to have this when I die, my kids are going to put it on my plate that he rolled up his sleeve when it mattered. So, and I think that is a great way to conclude. I'm going to end my talk, and then I'd be happy to take any questions.
Wonderful, wonderful, wonderful. Thank you so much, Dr. Swaminathan. It was really good to hear about the wonderful studies that's going on at Research with the Heart. And it was really, really enlightening to get a little bit more information about the J&J shot. I know in my community and my family, I've had people who actually got that shot, and then they called me hysterically crying and trying to get me to diagnose them with something that I didn't know anything about. So it's really good to be able to know different things that, the media doesn't exactly explain the way you just explained it. So I really appreciate that. And anyone who's on right now, feel free to submit questions. We do have 2 questions that came ahead of the talk. And the first question is, does vaccines work with the new strains? Now, I know you touched on the strains a little bit, but this is a really important question that a lot of people tend to have. And the second part of that is, if not, do we need to find new vaccines for each strain?
So I think very relevant questions. I believe that the vaccines do retain varying degrees of efficacy depending on the variant. And there's multiple different things that evolve over time. So what do I mean by that? In the UK, the variant that we talked about, Pfizer and Moderna have concluded based on their studies that the vaccine remains, the efficacy is not impacted. Over time, as the virus changes more and more, it is possible that if it accumulates enough changes in the virus and the spike protein changes enough, that maybe the effectiveness will reduce. I suspect that's what is happening with the South African variant. That being said, we are starting with a very high level. And what do I mean by that? In a good year, the flu shot is maybe 50, 60% effective. And that's in a good year. Most years, it's like 30, 40% effective. But it still works in preventing severe flu for the most part, right? So when we start with Moderna and Pfizer that are like at 95%, even when it drops to like, maybe 85 or 70, it's still way above what we would expect for effectiveness of the vaccine. So I still believe they retain efficacy. That being said, we never want to be caught sleeping at the wheel. So I think we are doing the right thing by trying to engineer these vaccines. And that's the beauty of the messenger RNA. You can tweak it quite easily to come up with a new vaccine strain for possible booster. And that's what they are studying to see if we really need that over time.
Awesome, awesome, awesome. The next question is, how will I know when I need the booster shot?
We need to do the clinical trials and then come up with guidance for that. I think that's a great question. At the current time, there is no evidence to suggest that you need a booster shot.
Okay.
For now.
Okay. Another question is, what happened to the people who were in the Moderna clinical trial? Is it ongoing or did it stop since the public has been receiving the vaccine?
So the clinical trial, so if you remember, in the clinical trial, 50% of the group got the active vaccine and the other 50% got the placebo. Once we proved that the vaccine was effective, it was unethical to follow people on the placebo. So everybody was unblinded, and everyone who received the placebo was given the opportunity to get the vaccine, because that is the right thing to do and the most ethical thing to do. Okay. With regard to follow-up, we continue to follow participants for a longer time to ensure durability of the safety of the immune response, meaning will it be as effective, 2 years down the road, and will it continue to be as safe? Those are things we continue to follow for these participants.
Okay, great. It looks like we have no more questions. So I will go on to say thank you, Dr. Swaminathan, for taking the time out to do this wonderful talk for Rutgers Day. And I would like to thank everyone affiliated with Rutgers and the university for giving our team and our site an opportunity to participate in such a joyous and celebratory day. We hope you got a lot out of this talk. Be sure to follow us on Facebook and Instagram at Research with a Heart. And we're also newly on Twitter, RWAH And stay tuned. We'll also be uploading this talk to our YouTube page. When it's available, we'll let everyone know. And I'm sure the Rutgers Day family will let everyone know as well on Facebook so that we can watch it later on. Thank you so much and enjoy the rest of your Rutgers Day.
Thank you so much. Enjoy. And get vaccinated.
And wear your mask still.
Yes. Oh, yes. Can I just add one quick thing?
Yes.
I remembered. So I read something about how do I know when I'm safe? 2 of 3 rules: outdoor, mask, or social distance. Take any 2 of those 3.
Okay. Yes. So no parties without masks.
Oh, parties? Oh, well, I'm a conservative person. I don't do parties. Not yet.
Be safe, everyone. Thank you so much. And we hope to see you all soon. Bye.
Thank you. Bye-bye.